And octopus consciousness is a really, really heavy promissory note, which I'm going to beg off of rather quickly. But what I really want to do is give you the notion, or at least my rationale for choosing octopus, of all of the invertebrates, of all of the animals. And what I like about the octopus is that there's this demonstrated behavioral flexibility that really rivals some of the higher vertebrates, at least some of the higher vertebrates that I've studied. It's a sophisticated nervous system that really is nevertheless a little less complex in its organization than that of the vertebrates. There are some functionally convergent aspects of its nervous system, its nervous system function. It does have LTP, but that's not really that unusual, as you'll see. You see this in a lot of invertebrates. You see this in a lot of animals. And finally, I think, at least to me, the diversity of the behaviors among octopus species really suggest some differences in neural properties that were driven by selection pressures in an environmental context. And this is an idea that will get me into my next pitch, which is for Jennifer Mather's talk, which is a week from Sunday. And Jennifer is going to give you a really, really good and compelling story about a possible ecological explanation for the possible origins of consciousness in the cephalopods. All right? So that's my pitch. Now, I want to give you a roadmap of how I'm going to go today. So I'm going to start with a working definition of consciousness, which won't take very long. I'm going to, it should take longer than it's going to take, is what I should say, because it's a pretty heavy bit of material. I am going to proceed then to talking a little bit about the structural and functional properties as I see it of consciousness, and then on to what I call invertebrate exceptionalism. Not American exceptionalism, invertebrate exceptionalism, okay? And then, again, to investigating consciousness in far-flung species more generally, okay? And then finally, I'm going to talk about an idea that I have, which may seem to some of you cockamamie, but it's a good, I think, platform for me to float this idea and get some interesting reactions. And I'm going to leave this up for a minute while I relate an allegory to you. It's an allegory that I think has resonance for people interested in evolution. So, the scene starts in heaven. There are three men who are right at the gates of heaven. St. Peter is looking down at them, and he says, well, gentlemen, you're all here for a good reason, obviously. You've departed. Your mortal coil is unwound. You're here. I'm going to let you in, but I really need to know how you got here. So what about you? So this guy's a businessman looking rather disheveled and hot and bothered. And he says, well, I was getting off of work late one night, and I had this suspicion that my wife was having an affair. And it just got into my head. I rushed home on the subway. It was sweltering out. I was panicked. I was angry. I get into my apartment. I immediately see that the door is sort of swung open on the balcony. So I go out, and the first thing I see on the fire escape is this guy. And he gets me so mad. I run to the refrigerator, and I get this burst of adrenaline. I pick up the refrigerator, and I drop it on him, and then I die of a heart attack. And then St. Peter says, all right, you're in. Yeah, that's good. Second guy, what's your story? Second guy says, well, I had a hard day at work. I'm in this 5-story walk-up. I come home. It's really hot. I have no air conditioning. Go out on the fire escape. I'm minding my own business, and this refrigerator drops in my head. And then he turns to the third guy, and he says, St. Peter says, okay, what's your story? Well, I was sitting in this refrigerator minding my own business. Okay, so it sounds like a very strange parable, right? But the idea is that evolution is something that we can look back ex post facto upon and make these wonderful stories about, but truly and fairly and honestly, as empirical scientists, we have limited amounts to go on, even though the stories are very useful for us to spin, because we can test aspects of the stories. And I'm talking about macroevolution here. I'm not talking about the evolution of bacteria, because that's absolutely very clear. You can watch all of that happen. You can watch a lot of evolution happen. But I'm talking about all the big stuff, like cephalopods diverging from the line that a line that split off and also led to what later became vertebrates. So there's my little tale, kind of a cautionary tale in some sense. Now I want to show you this to give you an idea. This is a very well-trodden video that has appeared on YouTube for quite a number of years, and Jennifer Mather and a few other people who are interested in cephalopods will be familiar with it. It's a little dramatic, but I'm just going to keep quiet and let you watch it for a minute. And I apologize if some of you have seen this already. All right, now here comes the money shots. I want you to watch everything in reverse and slow-mo. Is that cool or what? All right, so that bit of movie magic was captured by Roger Hamlin, who reports that he came up screaming when he actually took that video. He could barely contain himself. People thought he was having a diving accident at the time. And so he was an incredibly lucky guy, as I'm sure he'll report, to get that video. But what it speaks to is this amazing sensory motor capability, among others, in a particular cephalopod, in this case a common octopus, but in a lot of cephalopods as well. So this color change aspect, which I'll get into just a little bit later. But I want to kind of give you the richness and also a feel for the alien countenance of this animal to begin with. So let's start with a working definition of consciousness, in my mind. And I was influenced by another Edelman, a more famous Edelman, which I'll talk about on the next slide, who I'll talk about on the next slide. But I see consciousness, or at least what I refer to as primary or sensory consciousness, as the stitching together or binding, which is a very familiar word, as you all know, in consciousness research, of many sensory threads into a coherent, unified world scene. And the persistence of that unitary scene in memory. That, to me, is sufficient for primary consciousness. That also lets in, or at least allows the possibility of conscious states in a fairly wide variety of non-human animals, I believe. Okay? And the question of how this binding actually happens, I'm not going to get into today so much. I want to provoke you and get you to ask your own questions more than I want to answer that question. But in any case, I will make a key distinction about what I'm about to talk about. So I just mentioned sensory or primary consciousness. That is what I'm going to speak about today. And when I talk about that, I'm literally talking about living in the moment. I'm talking about this presence of a multimodal scene, and in deference to my father, who coined the notion of the remembered present, this idea of essentially living in the present, not having explicit access or recall to the past, and maybe some access to the future in a limited way in terms of planning, but nothing in consciousness necessarily. So you're living in the present, and when you're remembering, what you're doing is you're recategorizing the past or those dynamic interactions that stand in for the past in terms of your present experience. And this also sort of links to what William James called the specious present. William James was quite ahead of his time, and of course famous for pushing the idea that consciousness is a process and not a thing, which is a very important idea, in fact. Okay? And I'm distinguishing what I'm going to talk about today from something I'm not going to talk about today, which is what I call higher order consciousness. And this is the idea of a frame of reference that can encompass past, present, and future, conscious states that can actually reify the contents of primary consciousness as objects, okay? A sense of self, a true sense of self, an explicit construction of past and future scenes, okay? And again, I'm going to sort of steer a little clear of that, even though I am not in disbelief at all that certain animals are capable of some form of higher order consciousness. Just going to not quite be on my radar today. So now we're going to sort of go into a little bit of what I see as the structural and functional properties of consciousness. So some basic criteria, okay? The three big ones for me are these three ideas. First of all, that brain regions that function like thalamus and cortex and the connections between the two what we call, or what some people have called recurrent or reentrant signaling between thalamus and cortex. That's a very important bit of circuitry, I believe, to the conscious process. And the reason I say that is mostly from the human example. When we see humans that have been horribly damaged in some way or have basically an ablation of the central nuclei of their thalamus or have the thalamic link completely cut from cortex, just not likely they're going to ever regain consciousness again. And there's a lot in the literature there. I'm also talking about a widespread firing of neurons, of dynamic and fairly widespread neural activity across cortex, very low amplitude, but again widespread, that resembles something that we observe during the human consciousness, the human conscious state. And the literature is rife with examples of these sorts of things for the human case. Not as much for non-human primates, but there is some material there, and virtually almost nothing at all in other animals. Finally, the ability to make sophisticated discriminations, which would suggest some sort of a reciprocal connection between perception and memory. And that perception and memory aspect links right back to that thalamocortical and corticothalamic reentrant signaling. Okay? So this is just a laundry list. I'm not going to go into great detail of some of the properties that I would suggest that we can sort of assay for in conscious states or putatively conscious states, mostly in humans, but in many cases in non-human animals. Again, I won't go into all of these, but stability of conscious contents is a very, very important aspect, something that needs explanation. Allocentricity, the idea of sort of attending to others or having other objects, other people, other animals. Sensory binding, of course, as I mentioned before, and all of these others, and then some sort of an internal consistency, among other things. Okay? And so here we have sort of a depiction of sort of some of the limits of our functional neuroanatomical understanding of the hardware underlying conscious states. Of course, there's a lot more to it than this. I've sort of simplified things. But in humans, we often think of the links, and most of these reentrant, between cortex, thalamus, and the basal ganglia, which might be part of something you would refer to as value systems, systems that give the world sort of valence or salience, okay, or that point to certain things as significant and reinforce them over time and reify them as conscious contents. And something similar in birds. We're beginning to find out that birds, in fact, aren't very far from us in terms of their brain structure and functional anatomy. They're quite close, actually. And embryologically, you can show that almost nearly all structures you find in mammalian brains, you can also essentially find a version in bird brains, and there is a direct evolutionary relationship there. And I think most bird anatomists would agree with that now. And finally, the mystery of cephalopods and octopus in particular. We just don't know, is the short answer. And I'm not sure exactly how we'd assay for such things as reentrant connectivity and functionally equivalent analogs of thalamus. Okay? So here's an interesting question. If we're going to go into the world of invertebrates, we have to ask, have similar properties underlying sophisticated functions and behaviors emerged in nervous systems like that of the octopus, which are radically different from those of the vertebrates? And I think, and the course of the rest of my talk is going to deal with one sensory faculty, vision, that I think may offer a clue and perhaps an opportunity, maybe for me, but certainly perhaps for other people as well. And so when I talk about vision, I'm actually talking about something like distance vision. I'm talking about acuity at great distances, the ability to compose a scene, if you're talking about processing, that involves a great deal of detail from afar. In other words, you're very far from that predator, you're very far from that prey species. Okay? And I'm going to talk about this distinction, or the importance of that in a second. Okay? So if we look at vertebrate eyes, it's a pretty foregone conclusion. We know anatomically kind of what they're all about. They're these liquid-filled single compartment chambers containing a retina. We have focusing lenses by virtue, not really so camera-like in our case, by virtue of ciliary muscles, which can distort the lens, and we do things with our cornea that you don't see in invertebrates. And we have photoreceptors embedded within a retinal sheet, and that's fine and good. We all understand that. And in the case of vertebrates, we know that there are retinal ganglia. There's processing of what is coming into your eye right at the retina. Okay, and we know that there are input fibers that are going into the brain. There's the lateral geniculate, the thalamic relay, leading first to primary visual cortex, and then eventually splitting off and into higher visual areas, V2, MT, et cetera, that are involved in parsing out motion, contrast, color, and a wide variety of other visual aspects. But when you get to invertebrates, no such luck there. All of these, in the case of cephalopods, we can say they're there pretty much, although the focusing lens is more like a true camera. The muscles move the lens in and out and change the focal length between the lens and the retina. It's a little different than a vertebrate. Okay? So in the case of these animals, we have an optic lobe. We really don't have a retinal ganglia. We have a direct connection from the retinal sheet into the brain of the animal. A thalamic relay, we just don't know. As for rest of vision, damned if I know, but I'd like to know, and I hope at some point to find out through some good electrophysiology. So we have this problem when we move from the vertebrates to the invertebrates, and we're in particular interested in something like consciousness, which is what levels are of interest to us, or I should say more practically speaking, where should we start realistically, as opposed to spending the next 3 decades of our lives perhaps wasting time at the wrong level of resolution, which is always a possibility. We can combine and mix and match, assuming we have a lot of time, but we have to sort of choose our levels correctly, and you see various levels here. I won't dwell on these. But it's suffice to say that there's a lot that we understand about vertebrate and invertebrate neurobiology, even at the finest levels, that's been pretty well worked out. I call this the molecular transform of experience in the nervous system. And these are features that are all pretty common across vertebrates and invertebrates. Almost anything with a nervous system has these attributes, okay? And so I'm not going to dwell on this, but I'm just trying to make the point that even in animals as simple as this, you see these sort of attributes when you get to the finest levels of organization in their nervous system, the finest levels. At the molecular and cellular levels, you see these things, and they resemble one another across phyla and across evolutionary history. You have a very humble animal like C. elegans, this little worm. You also have sort of his famous animal, Aplysia, and here's the gill withdrawal reflex, and that's a sort of a classic reflex, and if you look at the neural circuitry behind that reflex, you see it looks like a very sort of basic wiring diagram. And in fact, you can knock out specific cells and you can get pretty specific effects very predictively by knocking out those cells. And again, no such luck once you get a few levels up in terms of degree of organization and heterogeneity and complexity and all. Okay? But again, we know that there's a basis for, I guess, what you might call learning at the synapse, changes in the synapse, that's common to vertebrates and invertebrates, although that view wasn't always the case. It wasn't always considered to be that way. Prior to 1995, we considered all of the important things happening in invertebrates to be happening presynaptically. We expected that all of it in vertebrates was happening postsynaptically. And the modern view, which is probably the more correct view, is in fact a lot of the same and common properties molecularly are happening at the synapse of both vertebrates and invertebrates. So there's that commonality. And you have, again, phenomena like LTP, long-term potentiation, in an animal like an octopus, only it doesn't depend on NMDA in this particular case. So that's different. Okay? So there are all these mechanisms that are common to both invertebrates and vertebrates. Okay? So now I'm going to sort of get into what makes the octopus particularly exceptional. Okay? And so here's the particular species I work with right now out of necessity because I'm presently a Californian, and there are plenty of these critters right off the coast, and that's where we get them. This is Bimaculoides, the California two-spotted octopus. And it's not a very good picture, but if you look sort of between the palm of my hand and the eye, there's this blue spot, and they're very characteristic of these animals. And in general, when we look at cephalopods and we look at vertebrates, obviously they're very, very different. They have different brain organization, different developmental pathways as far as we can tell, certainly different body designs and locomotion, but there are a lot of similarities. Similar genes in some cases, many of the same neurotransmitters. Of course, there are very ancient neuromodulators like serotonin, which are in a lot of things. similar nerve cell types in some cases, although there's quite a bit of difference there. Functionally convergent vision, again, which I'm going to dwell on in a second, and some comparable learning and memory faculties. All right, so let's get into a little bit about anatomy. And so, as I alluded to before, we can talk about structural and even functional homology increasingly comfortably when we're talking about humans, say, versus birds, or mammals generally versus birds. There's a lot of homology here indicated by the colors you can see. Cortex versus pallium, pallial structure here. It used to be that all of this was considered more or less like striatum in birds, and that view fell by the wayside, thankfully, quite a long time ago. But when we get to animals like cephalopods, the neuroanatomy is really, really alien and very, very hard to understand. We have some names of structures. We even have a little bit of functional anatomy worked out and function worked out for some of the so-called higher structures in the brains of certain soft-bodied or coleoid cephalopods, as well as nautiloids to some extent too, the hard-bodied cephalopod. So here's sort of a quick breakdown of the octopus nervous system. All right. So what you see is a very strange nervous system indeed, because it's parallel distributed. All right? More than half of the nervous system is essentially splayed out in the arms of the animal. These little knobs you see in each arm are ganglia, and in the common octopus they contain between 20 and 25 million cells each. That's a lot. In the central brain, you're talking about something on the order of 200 million cells. At least that's the latest estimate we get. But you can see how strange it is. And in many ways, the functionality of the motor system, or at least of the arms and arm movement, is sort of somewhat offloaded to the arms in terms of processing. There's still executive functioning coming from that central brain between the eyes, but a lot of motor programs that we would expect, say, in central pattern generation in the spinal cord of vertebrates are present in the arms of these animals in that ganglia. Something along those lines, although, you know, you cut off a leg, and it's not as if my leg is going to start executing a walking movement by itself, even if you jolt it with 10,000 volts. It's not going to happen. But with an octopus, you do get some interesting stereotype patterns of motion. So they're very alien in that sense. So just a few factoids. You're talking about roughly 500 million neurons in the nervous system of a common octopus. That's a lot. The central nervous system is, again, 200 million neurons or so. The central brain, to give it scale, is 200 to 10,000 times as large in terms of the number of neurons as you find in the brains of the bee and the marine snail, which is rather extraordinary. It's not even close when it comes to cell number in terms of other vertebrates relative to the cephalopods. And so a lot of this work, functional neuroanatomy was a great interest of this fellow here, J.Z. Young, who toiled away in Naples at the Stazione Zoologica Marine Station for decades, in fact. And he did quite incredible journeyman work trying to figure out what was going on in terms of, for example, vision and touch in these animals, because these are the two of the preeminent senses, of course, in the octopus. This is a cross-section of the octopus brain. The blue pathways are essentially visual. The red pathways are tactile. And Jay-Z also even made a stab at trying to understand integrative pathways, although nobody's quite cleared that one up yet. But he was on this very early. Unfortunately, his vigor and zeal about this, as well as that of Sutherland and people like Martin Wells, wasn't enough to sustain it to the present day. So essentially, this kind of neuroscience is not going on right now in a huge way in cephalopods. There's a little bit here and there, but nothing quite like this. And this is sort of more proof that we're dealing with sort of an alien organism in terms of brain organization. This is some of the few, these are some of the few findings that relate essentially to functional neuroanatomy that have come out recently. And this is from the lab of Benjamin Hockner working with Letizia Zullo. And they did both recording and stimulation of various areas, hoping to find some sort of representation, looking for maps of motor function in the brain. And I guess they were hoping that they might find something like topographical representation. And in fact, truth be told, they really didn't, at least at this level. They haven't found topographical mapping per se. But the interesting thing is that they used, when they did this, they did a tethered recording of single sites, one site at a time. And you can only imagine how many times they did this and how many animals they were doing this with. But they constructed this sort of collectively out of a whole bunch of animals, not simply one. And it's a little problematic in the sense that you really almost want an array. Anybody who's a hard and fast electrophysiologist says, well, how can you make a map unless you're recording from multiple areas at once, right? How are you going to know what's going on there? And so that's a quandary. It's great work, but it still leaves us this question. And the question relates to the problem of embodiment. We're dealing with an alien creature which leaves us scratching our heads. We have no comparators in the vertebrate world that will give us any clues as to sort of how the world is represented in these animals' brains, and in particular, how something like consciousness might be instantiated in such animals. So apologies to Thomas Nagel, but what is it like to be an octopus? And so now I will sort of briefly divert into one analog that's sort of interesting, a functional analog for hippocampus in terms of memory consolidation, conversion of short-term memory to long-term memory. Studies have been done, and ample work has been done in honeybees, in fruit flies, and Drosophila behavior people and Drosophila neuroscientists have teased out function that looks something like a hippocampus in the so-called mushroom bodies of the honeybee. And of course, these are anatomical features of brains you find in other insects as well. In the case of the cephalopods, well, there's this thing called the vertical lobe, and it was a great interest to J.Z. Young. And the reason we know that it might be doing something hippocampal-like is J.Z. did these amazing lesion experiments along with Martin Wells and other people in the 50s and 60s, where he just basically started cutting away aspects of the vertical lobe and testing the animals on certain kinds of memory tests, visual memory tests, tactile memory tests. So he was able to glean from his studies that, in fact, there is something going on in terms of effect on long-term memory or conversion to long-term memory in this vertical lobe. And if we assay the literature and we look across all the literature, when you look at learning memory tasks, octopuses in particular do pretty well in comparison to animals like rats. This isn't working real well. Rats and birds as well, which is pretty impressive. I'm going to show you one more thing, and it's rather controversial. I'll give you the Surgeon General's warning on this. This is not really accepted in the field as doctrinaire true. It's very controversial.
In the lab, the tank is divided into two by a glass wall.
Oh, sorry. I thought I was doing the right thing. Not doing the right thing. Here we go. So this was work that was done. It was based on work done 20 years ago by Graziano Fiorito and Pietro Scotto, published in Science.
In the lab, the tank is divided into two by a glass wall. A box with a crab in it is placed in one. Also in the tank is Candide, an octopus that was caught by a fisherman just that morning.
Am I okay for 10 minutes?
Candide sees the crab, but doesn't react. This is exactly what David Edelman expected.
No, it isn't, actually. No. I kind of shortened the video, so.
On the other side of the tank, the same box is given to an octopus that has spent a few days in the lab and has learned how to get into the multiple opening box. This octopus is called the trainee. It immediately starts to open the box to get at the prey.
Poor guy.
Candide watches the trainee, whose curiosity is aroused. The trainee isn't wasting its time. Candide is visibly excited and is clearly trying to understand what the octopus is doing. Finally, the trainee octopus opens the box.
Okay.
Now the glass panel between the two tanks is covered over, preventing the octopus from seeing.
Science is merciless.
Without hesitating, Candide grapples with the box and manages to get it open in barely a few seconds. He's observed and he's understood. The experiment is repeated dozens of times, and and the box is placed in different positions. The octopus always opens it the same side, using the same opening, showing that he didn't simply learn to imitate movement, but really understood what had to be done.
Okay, so now I'm going to give you a post facto set of caveats about this. It doesn't happen every time. Discovery Channel likes to say it happens every time. I wish I had known that's what they were going to say, because I would say it doesn't. It happens a lot, but it doesn't happen sufficiently to say, yes, these animals are capable of observational learning, and you can apply it across all of them, and it looks good in terms of statistics. But it looks interesting, is what it looks like. It's clear that some animals are amazingly capable of these kinds of things and may have nothing to do, as I had discussed with Jennifer Mather, with sociality at all. Because, as you might know, octopuses aren't really terribly social, at least what we know of them. And so I just wanted to wet your whistle for the possibilities there, because I do believe these animals are behaviorally rich. But I would say that there are some animals that are preternaturally talented. I would say there are a lot of animals that just sort of sit there, do that. Okay, so that's my Surgeon General's warning. So now I'm going to sort of get into the meat and potatoes of really what I want to get at here. So one of the onus that's on me, the thing that's the sort of Damocles over my head as a researcher who's interested in these questions, particularly in a far-flung species, is that I have to pull in order to make a convincing case. I don't have animals that are capable of giving accurate verbal report, although they can give behavioral report. That's something else I'll get into. But I have to pull in evidence, and sort of starting from the human framework, because I think it's a good place to start from. I need, in my mind, I need evidence of something functionally in terms of an interaction between neural structures that's going on that looks something like thalamocortical loops. Thalamocortical loops, corticothalamic loops, thalamic loops, corticocortical loops, just reentrant loops. I need the physiology. I need to actually see signatures. Now, I'd be interested in finding out whether those signatures resemble those of a conscious vertebrate, if the animal happens to be aware of a particular thing that I'm presenting, for example, on a screen. And I need to sort of look at sort of the widespread functional anatomy in terms of electrical activity in these animals. And we certainly don't yet have this in octopus. We have little bits and pieces in the vertebrates. Okay? And we're making a start. We did the first spike recordings in the brain of an octopus. We recorded from the optic lobe, and we basically showed the animal a blue light that's a conscious animal. It's knocked down a bit with magnesium chloride in its water and a little ethanol, but it's still awake, more or less, and it's attending to a blue light which blinks. And as the blue light blinks, of course, we're recording cell activity, and we got cells that were indeed sensitive to the blue LED. You know, they burst, and then they go off when the LED goes off, et cetera, et cetera. That's great. That is just a promissory note, obviously. That's not science. That's method. And what I'm going to show you is a variety of methods that I'm trying to build into a paradigm where I can explore the question of consciousness in a very substantive way, or at least in a way that will satisfy an audience like you that's accustomed to hearing about consciousness in vertebrates and in particular humans. Okay? So now I'm going to go really, really way out on the beam here in the next 7 minutes or so. And I'm going to talk about distance vision and consciousness and weave you a little evolutionary tale. Okay. So there's a more or less accepted assumption among evolutionary biologists who are sort of familiar with the Cambrian explosion in particular, but in general, evolutionary radiations of different animals. And one of the truisms that's emerged is that with mobility came the emergence, and this is all fairly obvious commonsensical stuff, although we can't replay the evolutionary experiment. With mobility came all kinds of elaborated predatory strategies. A sort of an arms race erupted between predator and prey species, and you got all kinds of innovations, including faster and more efficient locomotion, armor on certain animals, hard shells, peptide mimicry, which, by the way, you see in certain sea snails. It's really interesting. Which throw off predators by sending off a neuromodulatory system that immediately sort of defeats their appetitive drive, among other things. And finally, refinements in vision, which is going to be the subject of this last little bit. And so, of course, you know, if you're familiar with eyes, you know that eyes have been invented many, many times over the course of evolution, in parallel. There is no monophyletic origin for the eye, okay? And that even goes for the eye with the lens. And what I'm going to talk about in a second is the fact that octopuses have this eye that's very reminiscent of a vertebrate eye, all right? But for now, what I want to just simply tell you, suffice to say, that there are at least two major motifs in terms of eye design. One of them involves lenses, focusing lenses, and a single compartment. And you see that, among other things, in jumping spiders. You see it in octopuses. You see it in squid and other cephalopods generally, except for the nautilus. And the other eye motif, which is sort of the ommatidia, all of the little hexagonal, sort of particulations in the insect eye. And you see that in a variety of animals. But you also see some variations on this, which I won't go into. But in fact, this particular kind of shrimp probably has better vision than all insects do. But that's a different story for a different time. And so octopus vision gives me an opportunity. You have a familiar structure and function, I believe, in the eye of the octopus, suggestive of certain memory and integration substrates. And I think it's a great portal to exploit for investigating neural functions such as the conscious process. All right? So here to delve more deeply into the convergence, this is a little bit of a busy slide. I apologize for that. But basically the point is, if you look at an octopus eye versus a cod eye, there's a lot of convergence. There are a lot of similarities in terms of the morphology. They both have lenses. Both lenses are actually spherical, which is interesting. But the design is basically reminiscent of a vertebrate in many ways. When you look in detail at the design of the retina in the mammal versus the octopus, there are some distinct differences. In the case of the mammal, you have photoreceptors here, but light is actually entering and having to pass through layers of cells. It's actually passing through neuropile. And if you know about the human eye, for example, you know there's a fovea, there's a hole in the fovea. Everything has to pass around and back into the brain through that hole, to the lateral geniculate, eventually to the E1, right? Okay. No such problem with the octopus. An engineer may have designed, no, I don't really believe that. But the octopus, no problem. The photoreceptors just pass directly into the optic lobe. Light hits the photoreceptors directly. There's no retinal ganglia and none of that pesky processing to worry about. But that processing actually might be occurring somewhere in here, which is the first part of the brain that the connections from the eye hit, that the efferents from the eye hit, which is the optic lobe. And we started doing 3D reconstruction. This is an idea. We're sort of flying through this structure here. And if you look really carefully, you see these lightning bolts that are going radially down. Those are axons in all likelihood plunging into the optic lobe. But then you see collaterals crossing here. And it's a really interesting structure. But we have no idea functionally what's going on in terms of the actual processing. We just have some suspicion. So it's early in the game. But I will tell you, functionally, in terms of spatial acuity, octopuses do awfully well. This is just a measure of spatial acuity relative to essentially the body height of the animal to the size of the animal in some way. And if you look at where the octopuses are, look at where humans are, look at where falcons are, look at where octopuses are. Look at where the honeybee is. Look at where the blowfly is. That's not very good resolution up there. That's really good resolution down here. Pretty darn good. So distance vision is not, that's a pretty formidable thing, I think, in cephalopods generally, and in particular in the octopus. So now I have to engage in something that is sort of akin to invertebrate psychophysics to really unpack this, particularly on the road to understanding consciousness. Okay? So psychophysics obviously allows us to manipulate temporal and spatial properties of stimuli, and this is really important in vision, as you'll see, and perception ultimately. It allows us to monitor behavior for changes that indicate perceptual and integrative functions at work, and I'll show you some examples of that. And finally, it allows us, at some point, if we play our cards right and we combine it with good electrophysiology, to identify neural correlates of perception and integration. Okay. And we can monitor and perhaps investigate putative conscious states. So what do you show an octopus if you're showing octopus good video stuff? Well, you probably have to start with salience, and Jennifer Mather can speak more to the ecology of all of this, but it's very important to show them something that is sensible to them, that means something. So we can start with positive stimuli, like a crab, a yummy crab, or negative stimuli like moray eel. And these are sort of in the natural realm. This is sort of an array. These are in the artificial realm. So this is the graduate student that may come down and feed you every once in a while if you're an octopus in the basement of Stazione Zoologica. This is the white ball that you know comes down every time you're about to get fish, right? And eventually just the white ball comes down and you say, oh, wow, I'm going to get fish. I'm going to attack that white ball. Then there's this horrible red ball that comes down, and every time you approach it and you touch it, you get a 12 to 24 volt shock. And the third time around or fourth time around, you're backing off from it. So you can do conditioning in these animals, which is a very important tool. Okay? And now we have the problem of report, which in consciousness studies is actually pretty crucial, of course. You're not going to take my word for it if I come to tell you that I've recorded brain activity in this animal that looks conscious. You want to have some idea that the animal is giving me a report that indicates that it's aware of whatever I did show it in that particular instant. So the chromatophores are one way. You see on a young octopus, chromatophores expanding and contracting. There are these pigment-filled sacs on the skin of the animals that are responsible for its coloration, its patterning. Okay? So, and this is a video system we set up to explore these sorts of questions. I'm running a little late, but I'm going to speed it up. And so what you'll see is we hit a camera in a block. Here's a crab on a video. It's just a video screen. He's going to attack it. Now watch what he does. He backs off. He takes the measure of where the crab is again. And then he goes for it again. And he'll go for it a third time. And he'll keep going for it. Crab is a very strong stimulus. And so what are we going to do with these sorts of things, you ask? Well, for one thing, we can start to understand how octopus vision works, how octopuses sort of see the world. So this is a frame rate experiment. We started at a very high frame rate to test more or less the speed of octopus vision. And so here's what you just saw was the octopus attacking the screen, which is here. And you're seeing the picture of the screen. And if you look really carefully, that's the ghost of the octopus on the other side of the screen. Okay? So if we go down in frame rate, I guarantee you, you're not going to see any appreciable difference. There may be a slight latency difference, but the animals will still go for it. And I'm not going to dwell too much on this, but I'll suffice it to say I can keep on going down. All right, and now it's getting a little jumpy, and there's latency, and goes for it. Right, and now we're going to go down to 12 hertz. Okay, bam, and now here's where things get really interesting. At 4 hertz, now it's like Charlie Chaplin. It's worse than Charlie Chaplin. It's just a silent movie. Now you'll see, watch what happens in the middle here. See that arrow? That's where it basically changed color on half its body in response to what it was seeing. I'm not sure what that means, but it's a different response. And things are getting quite a bit ambiguous probably for it at this point. So finally, now to 0.5 hertz, and essentially, it's just a bunch of still images. And the behavior has largely gone away. Probably not sure what it's seeing at that point, right? So I present that by way of just saying that video is going to represent a very important tool for studying these sorts of things in these animals because we can manipulate it spatially and temporally. So we did a biological motion experiment. Are you guys familiar with the perception of biological motion? Just quickly, if you look at motion capture in Hollywood, when you mark people's joints with dots, and then you reduce them to dots against a black background, you can always recognize as a human another human walking. You can recognize a dog, you can recognize a horse, right? Can the octopus recognize a crab, for example? Silly question. So I'm giving you a sort of a toolkit to look at, and here's one example. We actually took some of the dots away. There were too many dots, but that's a dot crab. And the processor speed of my computer is really lousy, so I apologize for the fact that it was looking herky-jerky. But that's the idea behind that. And you present that relative to a control. In this case, it's a negative control. It's just randomly orbiting dots statistically, and I just cut it off. But you get the idea. Randomly orbiting dots. I'm going to show you this one movie here of a typical experiment. And sorry for the galaxy far, far away, blah, blah, blah. But the idea is you're going to see an order of presentations. You're going to see an empty tank. The octopus sees an empty tank. The octopus sees a real crab. Then the octopus sees an animated crab, which is a very photorealistic animated crab. Then he sees the dots. But it's not in that order. The reality of the experiment is we randomized it. They don't see that in that order. We're not conditioning them to to respond to the dots. We're always mixing the order of the stimuli. So here's the empty tank. And I'm sorry that is it still going? It stopped. Yeah. All right, well, anyway, that was just the empty tank. Here's the real crab. Still going for it. I don't know why it's doing that. Every time I push the laser, it seems to think that I'm stopping it, but I'm not going to push the laser anymore. Solve that problem. Okay. So I'll just point. Real crab. So he's gone to it already. And you'll see, in the next one, you'll see the digital crab. This is a digital animation. It's as photorealistic as we could get it at the time. It's still a little lame looking, but it's sufficient with a slight bit of latency to get the animal to respond. And the animal will go after it at some point as if it is. There you go. There's that. And again, I'm trying to show you a toolkit for future studies. I'm not trying to, this is not experimental data, obviously. This is the biological motion part. If you look really carefully, it's horrible contrast here, but it's great contrast for the octopus. There are blue dots, and it's a dot crab. And this particular animal will go for it. Yeah. And if you look in the next one, it's randomly orbiting dots. And at that point, it's sort of lost on the octopus. And I'll try to find it there. It might start to approach, but it's really not going to do very much. And in the interest of time, I'm going to go to the next. And the final thing that we're doing now, and again, we're trying to ply out the aspects of vision in these animals to understand it before. We have to crawl before we walk. In order to understand consciousness, I'm using vision as a portal, but in order to go there, I have to understand what's going on visually for this animal. Another experiment that we've done, and I have to mention that Jennifer Mather did a nice study about 20 years ago on visual navigation, mostly in the wild, but some laboratory-bound stuff. So here's where we set up something like a Barnes maze. Are you guys familiar with Barnes mazes, which are used with rats mostly? You have 18 holes, 20-degree increments, are on an arena, and basically the animal is dropped into the middle and eventually learns that the location of a particular hole is associated with either a distal landmark on the wall of that maze or a proximal landmark, something close by, proximal, something distal, far away. So this is one of our animals, and it's early in the game, but we are seeing animals, at least some animals, we're beginning to get a good significant set of animals that relate the visual landmark to the location of an escape hole, and they follow the landmark to the hole. And when you shift the landmark's position but keep the hole in the same place, the animals just go to the visual landmark. So they're associating that landmark. Doesn't sound like much, but when you do it over days, that implies a memory capacity, which is really quite extraordinary, which does relate to consciousness. So these ideas, I'm not even going to read these, but the importance of this is it suggests a depth of memory, a richness of memory that is very, very salient for anything relating to consciousness in these animals. And so I'm going to make the rudiments of an argument here. And this is the last slide. So animals with focusing lenses, single compartment focusing lenses, camera-like eyes, have developed more complex and plastic behaviors than other members of their phyla. Okay? Distance vision via focusing camera eyes has allowed animals more time to plan for and act upon far-off salient events or objects within a detailed visual scene. Okay? Distance vision evolved in tandem with neural circuitry for monitoring and prediction. And I don't think any of it so far is too much of a stretch, but it's on my head to demonstrate that sophistication in vision in the octopus. Distance vision paved the way for the elaboration of new kinds of memory, i.e. episodic memory, something hippocampal-like, okay? Because you have time now to play with. If you're a predator or prey species and something is far off, you have time to monitor and to start making decisions that you didn't have if you were a fruit fly and you can't see far off. You can only see when the hand is about to crush you. Okay? And so finally, distance vision also allowed for the generation of more detailed visual scenes and stuff that could be broken into constituent submodal properties. And this more or less would have gone along with the development of specialized neural architectures and a higher order means of integrating all of these things. So higher order dynamic maps, if you will. And that's what the holy grail is for, I think, all consciousness studies, but in particular for the octopus poignantly. And so in combination, once you have the integration of a unitary visual scene with this kind of memory to keep that scene persisting in the brain of that animal, and the emergence of something reentrant, that is something thalamocortical-like, that is more or less refreshing but is not, it's not recursive, it's reentrant, and we can go into that definition after the talk. You're basically setting up, I think, the requisites for sensory consciousness, and that's how I'm going to end the talk. Thank you very much, and I apologize for going over time.